In the winter of 1944, Nazi forces cut off food supplies to the occupied Netherlands. 4.5 million people were left to survive on whatever they could find. At the worst point, the daily ration dropped to under 500 calories. People ate tulip bulbs, they burned furniture for warmth. Tens of thousands died. Among those who survived were thousands of pregnant women, carrying children in bodies that were slowly starving. Those children were born small and malnourished. That part is not surprising. What happened next is. Decades later, researchers tracked down those children, now adults, and studied their health. They found that compared to people of the same age who hadn't been exposed to famine in the womb, these individuals had significantly higher rates of obesity, diabetes, and heart disease, even though they had eaten normally their entire lives. Their bodies had been programmed for scarcity before they were born. When food became plentiful, that programming worked against them. Remarkable, but not the strangest part. The strangest part is what happened to their children. The grandchildren of women who starved in 1944, people who were never hungry, born into a country that had fully recovered, also showed elevated rates of metabolic disease. Not because of their diet, not because of their lifestyle, because of something that had been passed down to them from a famine they never experienced. Something had been inherited that wasn't in the DNA. It has a name. It's called epigenetics, and it is one of the most important and most misunderstood discoveries in modern biology. So what is it exactly? To understand epigenetics, you first need to understand the problem it solves. Inside almost every cell in your body, there is a complete copy of your DNA. Your full genome, 3 billion letters of genetic code, is packed into each one of your 37 trillion cells. Every brain cell, every skin cell, every heart cell carries the same instructions. And yet those cells are completely different. A neuron looks nothing like a skin cell. A liver cell does completely different things from a lung cell. They have entirely different shapes, different lifespans, different functions. How can they all be running the same code and end up so different? The answer is that they're not running the same code. They're reading different parts of it. Every cell in your body uses only a fraction of your genome, the fraction it needs to do its specific job. A brain cell activates certain genes and ignores thousands of others. A skin cell does the opposite. The code is the same everywhere. What changes is which parts of the code are switched on. Epigenetics is the system that controls those switches. Picture your genome as a book, 3 billion letters long, containing the instructions for every protein your body can make. Now picture that book covered in tiny chemical tags, sticky notes, bookmarks, highlighters. Each one tells the cell reading the book, skip this page, read this one carefully, come back to this later. The text of the book never changes, but the instructions for how to read it change constantly. Those chemical tags are the epigenome, and unlike the DNA itself, which is essentially fixed from the moment of conception, the epigenome is fluid. It responds to signals. It changes based on what happens to you. Here is where it gets personal. Chronic stress leaves epigenetic marks. Prolonged exposure to stress hormones alters the chemical tags on genes involved in the stress response, making the body more reactive to stress in the future. In other words, sustained stress doesn't just affect how you feel today. It changes how your biology responds to stress for years, sometimes for life. What you eat changes your epigenome. Certain nutrients, found in leafy greens, eggs, and legumes, are directly involved in producing the chemical tags that regulate gene expression. A diet rich in these foods shifts the pattern of which genes are active. A diet poor in them shifts it differently. The same genome, fed 2 different diets, can behave in measurably different ways. Exercise, sleep, pollution, trauma, all of these leave marks on the epigenome, not on the DNA itself, but on the layer that decides how the DNA gets read. The most vivid demonstration of this did not come from humans, it came from mice. Researchers studying a particular group of mice discovered something odd. These mice carried a gene, called the agouti gene, that, when active, produced yellow fur, extreme obesity, and a high risk of cancer and diabetes. The mice were yellow, fat, and chronically ill. But when researchers changed what the pregnant mothers ate, adding specific nutrients to their diet before and during pregnancy, something unexpected happened. The offspring were born with brown fur. They were lean and healthy. They lived normal lifespans. Same gene, same DNA, completely different animal. The only thing that changed was the diet of the mother during pregnancy, which altered the epigenetic tags on the agouti gene, silencing it before the pups were even born. This is the same mechanism behind the Dutch hunger winter findings. The pregnant women who were starving in 1944 had their epigenomes altered by the famine, and those alterations were passed to their children, and in some cases to their grandchildren. The body, facing extreme scarcity, made epigenetic adjustments to survive. Then it passed those adjustments on as instructions for the next generation. The world is scarce, prepare accordingly. The world was no longer scarce. The instructions remained.
Now consider identical twins. Identical twins start with the same DNA, copied perfectly, letter for letter. At birth, their epigenomes are also nearly identical. But as their lives diverge, different diets, different stress levels, different relationships, different environments, their epigenetic marks begin to drift apart. By middle age, identical twins can have dramatically different patterns of gene expression. One may develop a disease the other never gets. One may age faster. Their cells running the same code are reading it differently, because the chemical tags on top of that code have been shaped by decades of different experiences. In one study, researchers examined twins who had been raised together until early adulthood, then lived separately. The pairs who had spent the most years apart showed the greatest epigenetic differences. The longer they lived different lives, the more different their biologies became, despite having the same genetic starting point. Scientists have now identified something called the epigenetic clock, a way of measuring biological age based on methylation patterns in the DNA. It turns out that some people's cells are biologically older than their calendar age suggests, and others are younger. The factors that accelerate the clock include chronic stress, trauma, poor sleep, and certain environmental exposures. The factors that slow it include exercise, stable relationships, and diet. Your epigenome is, in a very real sense, keeping score of how you live. This is why epigenetics is one of the most active areas of medical research right now. Because if epigenetic marks can cause disease, they can potentially be reversed. Unlike mutations in the DNA itself, which are permanent, epigenetic changes can, in principle, be undone. The tags can be added and removed. Several cancer treatments already work by targeting the enzymes that control methylation. Researchers are developing therapies that modify epigenetic marks linked to depression, addiction, and autoimmune disease. The idea that biology is destiny is giving way to something more complicated. Let's come back to where we started. The grandchildren of women who starved in 1944 carry something in their cells that was never in their DNA, a memory of a kind, of a winter their bodies never lived through. That is not a metaphor. It is a measurable biological reality, and it cuts both ways. If trauma and deprivation can leave heritable marks, so can safety and stability and care. The experiences we give to children are not just shaping who they become. Under certain conditions, they may be shaping who their children become. You did not choose your genome, but the genome you were born with is not a closed book. It is a book that is being read, highlighted, and annotated every day. By what you eat, by what you feel, by what happened to the people who came before you. The instructions were written before you arrived, but you have been editing them ever since.